everyone. So this video is about lipids. We're going to talk about lipid structure, what are the different types of lipids, and then how do those lipids come together to function inside the cell, and most importantly, how do they work to form membranes. So lipids are going to be different than the other macromolecules that we've talked about because unlike proteins and carbohydrates, to build them you don't take one piece and stick it to the next piece, to the next piece, to the next piece, and just go on. Instead, lipids are classified as lipids, or in this particular form, because they're all hydrophobic. That's because they're all made up of hydrocarbons. So those hydrocarbons are going to be just carbons and hydrogens all attached to each other. And so as a result, lipids will be hydrophobic. There are three major flavors of lipids. The first ones are triglycerides or fats. That's going to be things like fats, oils, waxes, stuff like that. The second one is going to be phospholipids, and those are going to be the most important type, the one that we're going to spend most of our time talking about, because they play such a critical feature in forming the membranes of the cell. And then the last one is going to be steroids, and those are involved in communication between cells and within cells. So here's a couple different types of lipids. This is the first question that's going to be on your handout for today. And I'd like you to look at these four different structures here and match them to the appropriate term. So steroid, saturated fat, unsaturated fat, and phospholipid. So that'll be the first question that I'm going to ask or to pose to you. And then in just a second, I'll tell you what each of those answers are. All right, so first let's talk about the steroid. The steroid is going to be this ring-shaped structure here. I don't know where my S went. All right, so that's going to be the steroid. This whole series of ring-shaped structures. This is characterized as a lipid because all of these are going to be composed of carbons and hydrogens. It's pretty much just carbon-hydrogen bonds with just an OH off the end. I'd like you to remember that at each of these different corners, there's going to be a carbon present there. All right, the saturated fatty acid is going to be this fatty acid up here at the top. Saturated fatty acids have single carbon bonds throughout their entire fatty acid chain. There are going to be no double bonds in a saturated fat. So this fat is going to be saturated with hydrogens in all of the positions around each and every one of these carbons. This lower fatty acid is going to be an unsaturated fatty acid. And you can tell that because it has a double bond right there in the middle. Because of that double bond, this saturated fat winds up having a bend or like a kink in its structure. And so it doesn't pack as well together in three-dimensional space. So unsaturated fatty acids tend to be liquid at room temperature. And then the last one is going to be this one up here, which is the phospholipid. The phospholipid is going to have two fatty acid tails. And a polar head group. So phospholipids wind up being amphipathic because they have part of them that's polar, this head group here, and then part of them that is nonpolar as well. So let's talk about fatty acids again briefly for just a second. So fatty acids can vary in the length or number of carbons that they have, and also the number and locations of any double bonds. So saturated fats are built like this, 3 fatty acids attached to a glycerol molecule, and all of these bonds along the entire length of the fat are going to be single carbon bonds. And then each carbon is going to be attached to hydrogens along its length as well. Because of that, these fatty acids are going to pack really nicely on top of each other like this. And as they pack really nicely, they will solidify and become solid at room temperature. All right, so things like butter, like this. So things like butter, these are going to be solid at room temperature. If you warm it up, it will melt, but at room temperature, it's going to be solid. Unsaturated fatty acids over here, they have got a double bond somewhere in their fatty acid length. And so as a result, they make a bent or a kinked structure. So they wind up not packing as well. And so because they don't pack as nicely and neatly, there's more space between the molecules as they move around. So they don't solidify particularly well. So then you get things like this, like olive oil, and that's going to be liquid at room temperature instead. Right, so we can do a quick comparison of different fats and fatty acids that you're going to be familiar with and talk about what they look like at room temperature versus the refrigerator, and then a prediction whether these are going to be saturated or unsaturated fatty acids. Okay, so let's start with butter. All right, so here's butter. At room temperature, we just talked about, it's solid, right? The same thing is going to be true in the refrigerator. It's going to be solid in the refrigerator as well. And so we predict based on that structure or the fact that it's solid at room temperature, we're going to predict that that is going to be a saturated fat. So this will be a saturated fatty acid. All right, what about coconut oil? So here's my coconut oil. Grabbed it out of the cabinet for you. All right, so this coconut oil at this room temperature, it's pretty chilly today, is also going to be solid. If I put it in the refrigerator, it would definitely also still stay solid. So we're going to predict that that is going to be saturated fat as well. Interestingly, if you leave coconut oil out on the counter and it's a pretty warm day, the coconut oil will turn liquid. So it melts at about body temperature, so about 37 degrees Celsius or so. Butter is going to have to be a bit warmer. You're actually going to have to heat it, put it in the microwave or on the stove or something like that. All right, now let's look at olive oil. So here's my olive oil. What is olive oil like at room temperature? Well, you can see here that it's liquid. What happens if you take olive oil and put it in the refrigerator, though? I don't know if you've ever done it, but it's really fascinating. So here I made this salad dressing with olive oil, and you can see that it's got this like chunky solid layer as it, so you can sort of see like there's a little bit of movement to it, but it's still a pretty solid mass because it solidifies in the refrigerator. All right, so it turns out this is going to be an unsaturated fat because it's liquid at room temperature, but it does solidify at pretty moderate temperatures. All right, and then the last one will be something like corn oil or vegetable oil. All right, so here's my vegetable oil. That's going to be liquid at room temperature, and if I were to take that and put it in the refrigerator, it would also be liquid. All right, so that's going to be another example of an unsaturated fat. The comparison that we'd like to do based on that is we can actually look at what the most common fatty acid is for each of these and do some comparisons about what that means for the structure of that fat. All right, so here we've got butter. The most common fatty acid is palmitic acid, which is 16 carbons long, and it's a saturated fat. Coconut oil is also a saturated fat, but it's only 12 carbons long. Because it's shorter, when those fatty acids go and line up and start to pack, the shorter length of that fatty acid means that they don't stick together quite as well. There's a little bit less hydrophobic interactions holding them together, a little bit less van der Waals interactions holding them together. And so the coconut oil is going to melt at lower temperatures than the butter is because they're shorter. All right, so I'm going to say melt right there. Olive oil and corn oil are both unsaturated fats. And I picked these two because they're actually the same length. So length doesn't make a difference here. But if you remember, olive oil with its most common fatty acid, oleic acid, that turns solid in the refrigerator, while corn oil does not. The reason is that olive oil just has one double bond right in that position, and corn oil has two. So corn oil is going to be a polyunsaturated fat, whereas olive oil is going to be a monounsaturated fat. because corn oil has more double bonds, it's got more bends and kinks and stuff in it, so it packs even less well than olive oil does. All right, so it turns out that the number of carbons matters. So the number of carbons makes a difference, and also the number of double bonds makes a difference as well for how solid or liquid each of these different fatty acids are. Right, the second type of lipid that I want to talk about are phospholipids. All right, so phospholipids are composed of two fatty acids. You can see those two fatty acids right down here. Here's their structure. Again, they're going to be primarily hydrophobic because they're made of just carbons and hydrogens. So this is going to be nonpolar. And then it's going to have one head group, which is this group up here, and this part is going to be polar. As a result, these phospholipids are called amphipathic. Amphi for both, and pathic for two parts, basically. So amphi, both hydrophilic and hydrophobic, and so they've got those two parts to them. Often we draw phospholipids looking like this model over here in the side where there's a circle with two little sticks coming off of it. We'll call the hydrophilic part a hydrophilic head. That's going to interact well with water. And then we have hydrophobic tails down at the bottom. What makes phospholipids so important is that they make these phospholipid membranes. So these phospholipid membranes can occur because the hydrophilic heads go towards the outside of the membrane and the hydrophobic tails point in towards the middle. So the hydrophobic tails are excluded from water and the hydrophilic heads are interacting with water. All right, that allows the cell to create a border or a boundary. The hydrophilic tails basically serve as a wall controlling what can go through and what cannot and gets bounced off of the membrane. All right, we're going to talk a lot more about that when we talk about membrane function and membrane transport, but this is what makes that membrane so important, is the fact that those hydrophobic tails can form a wall or form a barrier. All right, the last group of lipids are called steroids. So steroids are nonpolar structures, and they've got rings of carbon with hydrogens attached to them. This steroid here is cholesterol, and it's the precursor of other steroid hormones like estrogen and testosterone. And it's also an important component of cell membranes in animal cells. So not all cells have them. All right, these don't look anything like those fatty acids that we talked about before, but because it's made of primarily carbon and hydrogen, it classifies as a lipid. All right. So steroids and hormones have lots of important purposes inside the cell. They're used to make other hormones and vitamin D, and they're needed to regulate membrane fluidity. I think of steroids like the Goldilocks model, like the Goldilocks molecule. So you know the story of Goldilocks and the three bears, right? So she goes to the bear's house and she tries the dad's porridge and it's too hot. And she tries the mom's porridge and it's too cold. And then she tries baby bear's porridge and it's just right. Goldilocks likes things just right, just like cholesterol. So cholesterol sticks itself into the membrane in different places. And by doing so, it prevents the saturated fats from packing so tightly that it makes like a solid wall on the membrane. And so it forces them to move around and wiggle a little bit. It also takes the unsaturated fats, which are really movey and wiggly like this, and it packs in between them and sort of slows them down so that they don't become too liquid at the same time. So it helps to balance or buffer what's happening inside the membrane. So here's a little model of what is happening on the cell membrane. So down below us, we can see the cell membrane here, and you can see all the phospholipids moving and bobbing and moving around. And then there's a section here called the lipid raft, and that is a denser area of lipids. So you can see the cholesterol molecules packed inside of that lipid raft. And what I'd like you to note right here is that, do you see how each of these phospholipid tails look really stiff and straight? So these ones right here are going to be saturated fats or saturated fatty acids. And in fact, they're also just a little bit longer than the unsaturated fatty acids that make up the rest of the membrane over here. All right, so in the rest of the membrane, these are going to be unsaturated fatty acids, and here in the middle are the saturated ones. Now, when I turn the video back on, what I want you to notice is that the unsaturated fats move around a lot, and then the saturated fats hold pretty still and don't move very much. The orangey bits inside are the cholesterol molecules. So they help keep this saturated fat from becoming too rigid, so it still moves just a little bit. All right, so watch how these guys move. So with that, were you able to see how the membrane moves differently when the saturated fatty acids are there as compared to the unsaturated fatty acids? And then again, what role the cholesterol plays in helping to hold that membrane together correctly, and so that it doesn't become too rigid or too solid. All right, so here's another image where you can see the cholesterol packing here inside of the membrane and helping to regulate that fluidity. All right, so the next two questions for you are, which makes more stable packed structures, long or short fatty acids, and why? And what gives phospholipids the special ability to form membranes? All right, so I'll give you just a second or two to answer those in your video, and then we'll come back and talk about them. All right, so for this question, which one makes more stable packed structures, long or short? It's going to be the long ones, because the long ones have a greater distance over their length in order to have good solid interactions, whereas short fatty acids don't stick together as well, and so they're easier to pull apart. Think of like two pieces of Velcro. If you have two big long pieces of Velcro and you try to rip them apart, you have to go and separate the whole thing. But if you just have two short pieces of Velcro, you can rip them apart really easily. So long ones, because they can have better and stronger interactions between them. All right, what about phospholipids? What gives them their special ability to form membranes? So it turns out it's the fact that they're amphipathic. So here's our phospholipid. It has a polar head and then nonpolar tails. The polar head is going to interact well with water. And because of that, they can all line up next to each other like this. As a bilayer with all of the hydrophobic stuff in the middle. All right, so they can make really good membranes because they're amphipathic. They can hide their hydrophobic tails in the middle and then have their hydrophilic heads interacting with water on the outside. Right, so let's talk about the cell membrane or the plasma membrane for just a second. All right, so the cell membrane or the plasma membrane, its job is to separate the cell from its surroundings or the area around it. And it's made of phospholipids and steroids and proteins. And there are actually some carbohydrates involved as well. The most important feature of the plasma membrane is that it has selective permeability. That means that it allows some things to cross, but not other things. All right, so there are two words that come up when we talk about membranes. One is fluidity, the other one is permeability. And although they're related, they're not exactly the same thing. So fluidity is how much movement do you have in the membranes. So remember the phospholipid bilayer in the little animation where you had the unsaturated fats moving a lot back and forth and the saturated fats not moving? That's fluidity. So how much movement is there within the membrane? Permeability is something very different. Permeability is a measure of how much can cross the membrane. So if you've got your membrane like this, permeability is what are the things that can go back and forth and cross over that membrane. So they're not the same thing. And so one key is going to be to keep those two terms or keep those two ideas apart. And so here's an image of what that membrane is going to look like, or the fluid mosaic model of the membrane. It's called the fluid mosaic model because it's fluid. The membrane itself is liquid and can move back and forth, and all of the different phospholipids move around. It's called a mosaic because it's made up of a collection of different things. So it's got carbohydrates in it, and it's got proteins in it, and it's got phospholipids in it, and these little hexagon-shaped things, it's got cholesterol in it as well. So it's a mosaic. It's made of a lot of different things. And then it's a model because this is what we think the membrane looks like. It turns out it's actually really hard to image exactly what the membrane looks like, but using the information that we have, we put it together to create this sort of image. All right, so let's talk about the different components or different parts of the membrane. So you have the phospholipids themselves. We've already talked about them. They have a hydrophilic head right here, and then hydrophobic tails coming off of them. That one didn't work very well. Oop. Like that. So those are going to be the phospholipids. We've also got steroids like cholesterol in the membrane, and that helps to regulate the packing of the membrane as well to keep it from getting too rigid or too solid. And then we have proteins. We have two different types of proteins. We have integral proteins that go all the way through the membrane, and then we have peripheral proteins, which are just around the outside and just around the edges of it. And then finally, we've got carbohydrates involved. The carbohydrates are these green things, these green bits up here. And those carbohydrates can either be attached to the protein, in which case we call them a glycoprotein. Glyco is that prefix related to sugars again, like glucose or glycogen or a glycosidic bond. So these are glycoproteins because they have a carbohydrate on the protein. And then we can also get glycolipids, which is a carbohydrate attached to some lipids as well, again with that same glyco prefix. Let's focus on talking about the membranes in more detail for just a minute. So there are two major types of proteins that are involved in the cell membrane. The first one are peripheral proteins. These peripheral proteins are proteins that are bound to the surface of the membrane. They don't actually go into the membrane themselves. They don't actually get embedded into the structure of the membrane. They're just around the edge. So this red protein down here is going to be an example of a peripheral protein. It's related to the membrane, but it's not actually inside. And then there are integral proteins, and integral proteins actually go into the hydrophobic part of the membrane. You can have two different flavors of integral proteins. So integral proteins comprise both of these guys as well. Transmembrane proteins are ones that are going to go all the way through the membrane. So this yellow one here, you can see how it has bits of protein on both sides of the membrane. So that can go all the way through. And then there are lipid-anchored proteins that just send a small bit of the protein into the membrane. So like this green one is going to be an example of a lipid-anchored protein. All right, so but both of these go into the hydrophobic core. Both of them play a role in being firmly attached into that surface. All right, so here we are on the inside of the cell looking at the animation again, and I did this because I want to focus on talking about some of the proteins in the membrane. So this purple protein in the membrane here, this is going to be an integral membrane protein because it's actually buried inside the structure of the protein, or inside the structure of the membrane itself. And in fact, it's going to be a transmembrane type of integral membrane proteins because the protein here goes all the way across the membrane and it spans the membrane multiple times. So this is going to be a transmembrane protein. Okay, super cool fun fact is that scientists actually know how many amino acids it takes to get across the membrane like this. And the answer is 13. So it takes 13 amino acids to get across the membrane. Would you expect those 13 amino acids to be hydrophilic amino acids or hydrophobic ones? Right. So they're going to be inside of an alpha helix here, and it's going to be 13 of them, hydrophilic or hydrophobic. You let me know. All right, so this is going to be an integral membrane protein here. And here's another example of an integral membrane protein. This one is going to be a lipid anchored protein. You can see that there's a little part of the protein that just goes part of the way into the membrane. And so that's going to be the lipid anchored portion of it. Most of the protein is on the outside. What's the function of these integral membrane proteins? And they get many different functions. Some of them might be transport, so they move molecules across the cell membrane, either in or out, and we'll talk a lot more about that when we talk about membrane transport. Some of them are enzymes and have some enzymatic activity to them as well. Sometimes they're signaling molecules, so they don't let material through the membrane, but they let ideas or information come through. Sometimes they're involved in cell-to-cell recognition, like this glycoprotein here. This glycoprotein has carbohydrates, as that little green part right there. And those carbohydrates can then serve as a little message to a neighboring cell identifying what the particular cell type is. Some of these proteins are involved in intracellular joining or connecting the two different cells to each other. And sometimes it attaches them to the extracellular matrix or all of the stuff that is around and outside of the cell, and it attaches and holds it in that particular position. Also some carbohydrates in the membrane as well, and membrane carbohydrates are generally surface recognition molecules. These carbohydrates can either be attached to lipids, which makes them glycolipids, or they can be attached to proteins, which makes them glycoproteins. And these carbohydrates have incredible amounts of variability. They're really different from one cell to the next and one person to the next. So in fact, when you think about blood cell types, you probably even know what blood type you are. That blood type, whether you're A, B, or O, is controlled by which glycoprotein you have on the surface of your red blood cells. So if you have the A glycoprotein on the surface of your red blood cells, you're a blood type A. If you have the B, you're a type B. If you have both, you're AB. And if you have neither of them, you're going to be a type O blood. That information gives signals to the cells about who you are, so that if you receive blood from somebody who isn't of the same blood type as you, your body's actually going to make an immune response against it, get rid of that blood. So it's really important in a blood transfusion to have that all matched up. I want you to take this image of the cell membrane right here and label it with all of these different parts or different components. Where are the carbohydrates, the glycolipids, phospholipids, things like that. And I'll post the answer key for you on Canvas so that you can see where all of those different parts are. Right, so if you have any questions, definitely bring them with you to our class Zoom meeting, and I will see you there. All right, have a great day. Bye.